Today I will be sharing two of my projects. These two might not be the latest ones, but they're kind of representative ones. So I've been working on spatial omics, brain images, and also a little bit of text data. I'm interested in text data, but it's not what I'm working extensively right now. So I'll be introducing one project in spatial transcriptomics, and it's on the high resolution technology of spatial transcriptomics. And the another one is on brain imaging, which is motivated by a phase 3 clinical trial data, and we proposed a novel mediation analysis model for this problem. So let me start with the first one. It's about spatial transcriptomics. So spatial transcriptomics and proteomics, they are elected as a method of year by the Nature community in year 2020 and 2024. So these are kind of important technologies well recognized by the field. And people tend to believe these are the next generation technology that will help us advancing the biology and also the treatment. So this is kind of a typical visualization of how the spatial transcriptomics data looks like. So on the right-hand side, you can see there's a slice of mouse embryo. It's kind of small, like it's 2 centimeter height and a little bit more than 1 millimeter wide. But there are really lots of information can be provided using this spatial transcriptomics technology. For example, if we zoom in, we can really see the differentially expressed genes. Like this particular blue gene is expressed in those particular cells in this area, and we can nicely see the structure. And if we do the cell segmentation, similarly, we can annotate the cell types. So basically, for each dot here, it represents a cell, and the color corresponding to its cell type. So for example, all the dark blue cells, they belong to the same cell type, and we can map them to the real location of this mouse embryo. So we can nicely see the tissue architecture on this tissue slice. So this really like the great part of this technology. We can no longer know we can not only know the gene expression, but we can also know their physical location. This means we can answer a lot more questions with this type of technology. For example, what's the tissue architecture, why this cancer is progressive, how the cancer cells it's communicating with the immune cells, etc. But this technology is new, it's comparatively new. So people are still developing new technologies to capture this type of data. We can see there are more and more and better technologies come out each year. So the number of technologies available, it's increasing. And there are also many public available data sets. People are they want to create atlas or large data sets so that researchers can work on to together push forward the field. So there are roughly 2 different types of this technology. One is called sequencing-based, the other is called imaging-based. For the sequencing-based technologies, stereo-seq and Visium, these are really popular, and they sequence a lot of lung tissue during COVID time. And for the imaging-based technology, geomics from NanoString, a company is quite popular, and definitely Zynium and Murfish from the Harvard team, and Zynium, which is a very popular and famous company right now, are getting more and more popular too. So this is about the data and the technology. So for this particular project, we're interested in working on those kind of new high resolution spatial transcriptomics. Here it's a slice. It's high resolution because if it zoom in, we can really see the details at cell level. For example, in this picture on the right, those white boundaries are the cell segmentation boundaries, and those dots with different colors are the expression of different genes. So we can really see those blue genes are expressed in those a few cells, and these green genes are expressed in these cells. Maybe they are another cell type, and those red genes are expressed in those larger cells. And we can also see the subcellular structure of the gene expression. For example, in this particular cell, the gray shadow in the middle actually highlights the cell nucleus. So we can see those red genes, they're kind of in the cytoplasmic area, right? So they're more expressed between the nucleus and the cell boundary. So we can really see some subcellular structure from this high resolution technology, and we want to make use of this information. But however, we are statisticians, we're interested in this topic, but how we can get our work started. We want to know how the data looks like, right? But actually the data for spatial transcriptomics quite nicely processed. So there, we mentioned there are two types of technology. We check out the sequencing-based first. For the sequencing-based technology, we basically have very tiny capture areas scattered in our tissue slice. So in this case, we highlight two cells in this cartoon. So there are many tiny capturing areas, and there are so many in the same cell even. And for each capture area, there are many probes. So those probes can stick to the molecules. And if we're sequencing on those probes, we can have, this probe also has a barcode area. So when we do the sequencing, we can sequencing the barcode on the probe, which tells the spatial location. And we can also sequencing the molecule that sticked to this particular probe. So it always have paired reads, the so-called pair reads. But at the end of the day, the data we have is two matrices. So for example, if we look at this particular capture area, let's call it spot number 2. In the first matrix, we will have a row, this second row corresponding to spot number 2. The first elements corresponding to first column, which is the number of counts of the first gene. So I think it's pretty neat. Like for this row, we have the counts of a number of genes. And in the other matrix, if it's checking out the same row, it tells us the coordinate, the X and Y coordinate of this particular spot. So this means we're checking out this row, we know this capture spot is at this location, and here's the gene expression across a number of genes on this spot. So this is basically how this data, process data looks like. There's another type of technology called imaging-based, where the data is kind of a similar, a slightly different flavor. So a cartoon is here. So it has many point clouds. So there are lots of points, but the points with different color. The color code different genes. For example, if we're checking out this row, it's a pink row. It means like all the pink dots, they corresponding to particular gene, and we record the location of all the pink dots. So which means all the purple dots here are the purple gene, all the green dots here are the another gene. So we have a 3-column matrix, the first 2 column corresponding to the location, and the third column corresponding to the gene name. This is equivalent to the 2-matrix structure, actually. But anyway, in both technology, we will have the gene expression information and also the spatial information. Usually one coded in one gene expression matrix and other coded in a location matrix. So I hope the data structure is not very confusing. So in this particular project, we are interested in developing a statistical tool for detecting subcellular features. Why we're interested in this? Because this data, this new technology enable the resolution at subcellular level, and also the studying the subcellular mRNA localization is important for understanding the cellular function and also disease etiology. For example, the mislocalization of mRNA, for example, if mRNA, it's expected to be localized in the nucleus, but in some particular cell, it's in the cytoplasmic, this may be related to some disease like cancer or some others. And also the compartmentalized translation, because mRNAs will be translated into proteins, right? This type of compartmentalized translation can help with the cells to do their functions. And this is also related to the microenvironment. For example, the immune cells and the cancer cells, they're talking to each other using particular proteins and RNAs like the ligand and receptors. So if we can work on, suppose these 2 genes, they're all sit at the boundary, they're talking to each other, these can help us better understanding if this microenvironments, for example, immune hot or immune cold. And this can be related to the treatment treatment outcome. So this is kind of the motivation why we think it's important to study the subcellular localization. But however, there's really a lack of good method for detecting the subcellular pattern. For example, this particular mRNA, it's in the cartoon, it's in the cell protrusion, and this is on the nuclear edge, and this one is probably on the ER. So we want to develop a method that can detect the patterns of at subcellular level. So this is the STAT method or model we worked on. It's called ELA. It's for both detecting if a gene has subcellular pattern and also for estimating its pattern. It's based on the non-homogeneous Poisson process, a spatial model. It's quite flexible. It can be applied to many cases. And by applying this method to some real data set, we also find some interesting findings, such as interpretable mRNA characteristics. So our method mainly consists of 3 steps. The first one is data preprocessing, because we just introduced the data we got as kind of the 2 matrix structure, but how we can process them to make them become the input of our proposed model. And the second step is fitting our model. And the third step is testing, like if this gene has the pattern and also have the pattern there for us to do more downstream validation. And let me introduce this step one by one. So for the first step is preprocessing. The very first thing we do is modality alignment, because the transcriptomics data, they usually come with staining images. And those staining images are usually more informative to provide the cell segmentation information. So we got cell segmentation information from the accompanied images. And this is a sequencing-based technology. One advantage is for the sequencing type, we can differentiate the mature and immature mRNA. So using the immature RNA, it's usually localized within nucleus. So this is basically, we have information to tell us where is the nucleus. So at the end of the day, this step is for us to get the nuclear center, where's the center of the nucleus, give us a landmark of a cell. And also where's the boundary of the cell, another landmark. And then we do cell registration. The thing here is we have many cells on our tissue slice, but our signal can be noisy. But how can we increase our confidence? It's if we repeatedly observe one pattern across many cells, then we have more confidence, right? If it's in the nucleus in one cell and the cell do 100 more, we are more confident to claim, okay, this pattern is correct. But the cells, they have different shapes. So what we do for this step is we use our landmark, one is the nuclear center, the other the cell boundary, and we compute for each spot or location, we compute 2 distances. One is this distance to the nuclear center, the other is to the boundary. And we divide the first distance by the total distance so that we get a number from 0 to 1, right? In this way, we kind of registered all the cells into a unit disk, where the center is just the 0 and the radius equals to 1. So this is the step, like how we can register all the cells into one template so that we can work on multiple cells in the same coordinate space. So we can see after the registration, we still keep the pattern to the landmark. For example, this particular gene in this original cell looks like in the cytoplasmic, and after registration is still kind of in the cytoplasmic, and so do the others. So the purpose of this step is really enable us to work on multiple cells at the same time. So this is basically our preprocessing step. We got many cells of different shape, and we register them all into a unit disk. What our input for our model is really the 1D distance. So the distance between 0 and 1, we just computed. So our model so far only models patterns in 1-dimensional space. So after computing this 1-dimensional distance, like how far this point is to the registered center, we can place those points on this 0, 1, one-dimensional interval, the intuition is, for example, if we look at those red points in the original cell, they're quite in the nucleus, right? And after registration, they're still near the center. If we put them in a one-dimensional interval, then they're here, they're quite close to 1, right? So suppose if we estimating a density on this 0 to 1 interval, we imagine the density curve goes down, right? So this is kind of the idea of this model. We want to estimate this pattern. If the density is high at 0, it's corresponding to a nuclear pattern. This is kind of the idea of our model. But anyway, here's the preprocessing step. And after the preprocessing step, we can feed our model in the one-dimensional space. So it's a non-homogeneous Poisson process model. How it works is, suppose we choose an interval from A to B, for example, 0.1 to 0.2. And this index I represent the ice cell. And this J, it's for the J's dot, J's point, and this particular cell. So if we count how many points of this gene in this interval, it follows a Poisson distribution. That's the idea of the non-homogeneous Poisson process. But the thing is, the intensity of this Poisson, it's an integration of this HPP intensity. This intensity varies from 0 to 1. So the intuition is because this is a function varies from 0 to 1. If we integrate it at different interval, then it gives a different value, which means the Poisson intensity are different. That's how we model the variation on this interval we just talked about. So we did a little bit decomposition of this K term because we said we want to borrow information, so we want them to share the same pattern across different cells. But we definitely want to adjust for cell level features, right? That makes sense. Different cells, they have their own thing. And this S term is because our cell is in 2D, but now we're model it in 1D. If we imagine it, we can imagine that when it close to the center, we draw a small circle, right? But when it goes to the boundary, the perimeter get longer. So we have more area when it goes outside, right? So that's why this SR equals 2 pi r, the length of the perimeter, to adjust for the area effect. It's just because we compress things from 2D to 1D. And this lambda r is K term that's shared across cells to model the pattern we want. So here's the model fitting part. After we propose the model, we want to estimate everything. The strategy we used here is we propose a number of kernels and we fit one kernel at a time. Why we do this, it's for numerical stability and also for we can do everything in parallel. Here we choose the beta kernels, and here we listed 22 beta kernels. So each of them, it's a PDF function of the beta distribution. Why we choose it, because it's kind of a natural density on the interval from 0 to 1, and it's quite flexible. Like if we adjust the hyperparameters, it can have varying different shapes that can be flexible to capture any real pattern. So we fit one kernel at a time. We allow a linear transformation to fit our unknown pattern. Of course, those two should be greater than 0 because we will not have any negative density function, right? So here for each kernel, we will have two unknown parameters. We estimate those. We find the optimal value of those two parameters using the ADAM algorithm, so just the optimization algorithm. In a special case, we are requested to add one more random variable to handle more over dispersion. So if we add one more random variable here, we will use the policy gradient to handle that. But the idea is we apply some optimization algorithm to find the optimal parameter value, so that for each kernel I, we will have its corresponding estimated parameters, and therefore also its estimated pattern function lambda, because it's computed from those three. And the next step is, remember, our goal is we want to know if this gene has pattern or not. This corresponding to hypothesis testing, right? So under the current model fitting setup, it's pretty straightforward. We're basically testing if beta is 0 or not. Because if beta is 0, then lambda becomes this alpha. which is constant, it corresponding to random pattern because there's no variation. If our beta is non-zero, then it means it has certain projection on this particular kernel. For example, this one, it means it has some pattern. So that's our hypothesis testing, beta 0 naught, and we use likelihood ratio test. So after this step, we will have one p-value for each kernel. Because we definitely want to summarize all the information captured across all the kernels. So we will use Cauchy combination to combine all of those p-values to one for one gene. But so far, we always talk about one gene. In the real data, we have many genes captured by the technology. So if we have many genes, we apply our method to one gene at a time. Then we need to adjust for the multiple testing issue. That's why we have the FDR control p-value if we have multiple genes. But the idea is we first have one p-value for one gene. So now we have one p-value for one gene, but how about the pattern? We still have one estimated pattern corresponding to one kernel, so we'll use the Bayesian model averaging to integrate all the estimated patterns together. So basically, at the end of the day, by applying our model or method, for each gene, we will have one p-value. So suppose here are three genes, like for each gene, we will have one p-value and we will also have its estimated pattern. For example, in this toy plot, this red gene is high at 0, corresponding to the nuclear center. So this red gene, it's a nucleus enriched gene. So that's kind of the idea of this model. So next we do some simulations. There are a few, but not many competing methods. For example, sprawl and Ventol. This one is a statistical method. This one, it's a random forest method. So this one don't have p-value. And some researchers, they just count the number of nucleus counts and the cytoplasmic counts and do a Wilcoxon rank sum test. So these are the competing methods. Overall, our method is the one that can be applied to most of the cases is flexible. And also it requires reasonable information because we don't need nuclear boundary segmentation, we just need a nuclear center. So this is a kind of idea of the competing method we have, not many. So our first set of simulation is now a simulation. The idea is we generate a bunch of data without signal. Then we will not expect our model detect anything, right? Because if our model detect anything, this is going to be false positive because the data we generate has no signal at all. So this is the idea of this simulation. So we generate the synthesized data from some real cell templates. We got quite some replications, and we compute our method with the existing method. For example, we find this pink method corresponding to Wilcoxon rank sum test, it had quite some false positives. But our method and the other existing statistical-based sprawl method works quite well in terms of control the false discovery rate. And then we do some alternative simulation. The idea is, suppose we generate some data with signal, then we will expect our method detect it, right? If it detects nothing, then that's called no power. So this another set of simulations for testing the power. So we have quite some settings with synthesized pattern. We can see our method colored in green had the highest power around all of these settings than the competing method. Also, although our method is in 1D only, so it's not expected to detect 2D patterns, but it actually can detect some, except for one pattern. Like if this two-dimensional pattern is completely uniform on a angular sector, this means it has pattern on 2D, but it has no pattern on 1D. So this is a limitation of method. We cannot detect this type of pattern just because we are 1D right now. But this pattern is actually not very commonly observed in the real world. So that's the idea of another set of simulation. We have great power and outperform the competing method, except for one case because we are 1D. And we can also see one favorable thing for our method is we can estimate the pattern, but while all the competing methods, they can only tell us if it's significant or not. It will not provide us how the pattern looks like, but our method can give us how the pattern looks like. So the dashed dark green curve is the true pattern. The light green curves are the estimated pattern, and the gray ones are the non-significant things. So we will not care about non-significant things, but we can see the significant estimates are quite accurate from the plot. Yeah. So we said our one advantage of our method is we can borrow information across multiple cells to increase our confidence and also have more power. But what if we just have one cell and we have good signal to noise ratio? So we also compared our method in the setting that we only have one cell with the existing method, and we can see if we only have one cell, if our signal to noise ratio is good, we can have good power without working on multiple cells as well. So this it's about this simulation setting. And we apply our method to several real data sets. Here I'll show two. The first one is the data set we show at the very beginning, the most embryo one. We focus on two cell types near the heart region. These are the myoblasts, which it's a type of immature muscle cells, and also the cardiomyocytes, which is a mature muscle cell. We did cell segmentation and we identify nuclear centers, and then we apply our method. So here are the estimated patterns. We clustered the lambda estimates, the significant lambdas from our model. We clustered those lambda using k-means into 5 pattern clusters. So we can pick representative genes in each cluster. For example, the first cluster we can pick, also there is our estimated pattern. The background is a density map showing how the expression looks like in the true data. And here we overlay 5 cells together to check out how the actual expression in 2D looks like. So we can see it's quite nucleus enriched in those 5 representative cells. And this also gives us the insights why we really need borrowing information across multiple cells, because it's really sparse. It's like we do need to overlay them together to see the pattern. So that's the result. And we also want to do some validation to check if our results from real data, because there's no ground truth anymore, to check if they make sense. So one thing we used is something we already know is the unspliced data, which are immature RNAs. They are more likely to sit inside the nucleus. So suppose we detected a gene, they are more enriched nucleus. It makes sense that it has more unspliced counts, right? Because if you look at this one and this one, those cartoons, more nucleus means higher proportion of unspliced genes. So when we compute the ratio, we find our nucleus genes, they have higher unspliced and spliced ratio, which makes sense. This is one validation. The other one is we kind of heard about the central dogma, more or less, I guess. So the DNAs, they transcribed into RNA inside the nucleus, and then they translate into protein, the cytoplasmic. So we find that longer genes, they are more likely to be enriched in the nucleus. There are some support in the literature. The hypothesis is longer genes, they need longer time to get transcribed. So cells kind of want to get them prepared and stock them in the nucleus, so that when I need them in the cytoplasmic, I can just export them. I don't need to like take time to transcribe them anymore. So that's the idea. So it's kind of makes sense. There are many long genes, they have high expression in the nucleus. So and similarly, we find the transcription factors, they are more likely to be localized in nucleus because they are expected to regulate the transcription. And we find the ribosomal related genes, they're more likely in the cytoplasmic because these are kind of the location they're expected to work at. So Yeah, this is about this data set. Another example is we work on another imaging-based Sickfish Plus embryonic fibroblast data set. This is cell line data, which means the spatial structure is not as fancy and all of those cells are basically fibroblasts. But the throughput, it's good. It means it has very nice signal-to-noise ratio. So we can observe it from this plot. So similarly, we estimate the patterns and clustering them into clusters, and we pick one representative for one cluster. And here in this data set, the signal is stronger so that we can visualize the pattern in one cell and we can check, okay, makes sense, this is a nuclear pattern. So that's about this data set. We did some similar validation about the gene length and about the transcription factor, and we found consistent findings to the previous data set. This kind of increased our confidence because across platform, across data set, we have the consistent findings. We also did some unique downstream analysis, which is interesting in this data set because this data has very good throughput, and we can actually identify the cell cycle phases of these fibroblasts. Then we can check the variation of the subcellular localization across the cell cycle, which is a pseudo time or a kind of time, like the cell, it's so from G1 to S and G to N phase along the time axis, we can find there are some dynamics in the subcellular localization. For example, this particular gene is not as much nucleus enriched in the G1 phase, but it becomes quite nucleus enriched in the M and G2 phase. So the dynamics of subcellular localization is so quite interesting because it's related to the regulations and functions of cells. Yeah, that's basically about the first project. So we developed a method and applied to quite some real data sets. Yeah. I'll skip the summary and introduce the second part. So I also work on brain imaging quite intensively. So there are brain imaging slightly different from spatial transcriptomics. It's a very mature technology, and it has been widely applied to the clinical and medical side. There are many modalities, but in this project, I actually work on the diffusion tensor imaging. So this project is motivated from a phase 3 clinical trial at St. Jude. It's a clinical trial about medulloblastoma, which is a very common brain tumor among children, and the tumor site is roughly at the back of the brain. So the standard therapy is radiation therapy. Like after we use surgery to remove the primary tumor, we apply radiation therapy to the cranial-spinal area and also apply some extra radiation therapy on the primary tumor site. So that's kind of the idea of the treatment. The key is they have different treatment groups. For the average risk group, they receive slightly lower amount of radiation. For the high risk, the radiation dose is higher. Higher dose means more side effects. So it makes sense because those children, they're really young. Some of them are less than 5 years old. So they will suffer from cognitive deficits. That's because their brain is still developing, but those radiation therapy can really affect the development of their brain. So we can see usually the high-risk kids, they have a lower cognitive outcome than the average group children. So our goal is for this particular disease, the good thing is the survival rate, it's above 90, sometimes even above 95%, which is really good. But some children, they need to go to special care because of their cognitive deficits, which is the not as good part. So our goal is how we can increase the quality of life of the cancer survivors. So our idea is we integrate the brain image into the analysis. So there are some existing studies show that treatment damage the brain, which totally makes sense. And the brain, it's associated with a cognitive outcome, which is quite expected because that's why the brain works for kind of. So our idea is we want to find the brain structure that is damaged by the treatment that affect the cognition, which means we kind of find a negative pathway that enabled by this damaged brain structure. So along this pathway, it pass some negative effect to the quality of life of the survivors. So our idea is what if we first identify this pathway, and if it block in the future, if we update the treatment, we block this pathway. In theory, we will give the children better quality of life because we block some negative thing. So that's the idea of this project. So this is a natural way of doing mediation analysis, right? So it's very natural idea to apply the mediation analysis. The only thing is we really have a high dimensional mediator, which is the brain, 3D brain image. So there are some existing work. There's some analysis on either of the pathway, either treatment on brain or brain on outcome. And there are some high dimensional mediation methods available too, but they're not really tailored for this case. So in our case, our dimension is really high because the resolution of the neuroimaging improves so much. And we have spatial sparsity because given the large p small n thing, we don't expect every pixel to be significant, and also it makes sense in the real case, not every brain region matters. We have spatial dependency because it's imaging type data, and we also hope to have a nice computational time because we don't want our code to run forever. So we propose a new model or a new prior, which is for doing high dimensional mediation model and with variable selection, also smoothing to address the sparsity and also the spatial dependency. So we follow the classical mediation analysis framework, which means the first step is we use the mediator, the high dimensional mediator as outcome, our univariate treatment as the predictor or covariate. So this outcome is high dimensional, this predictor is univariate, and we will have a high dimensional coefficient corresponding to the high dimensional outcome, which kind of makes sense. So M is mediator, X is the exposure. And the second step of the standard mediation analysis is we regress the outcome on both the mediator and the exposure. So the outcome, it's neurocognitive score, it's univariate. And here we have our high dimensional mediator where we want to integrate all the effects across all the voxels. So here we have integration, or we can use summation here because we summarize the effect all the voxels or pixels together. And here's our exposure variable, and the gamma is the so-called direct effect. So that's how our model looks like. And after proposing this model, we can have our mediation effect calculated, and we can also have our direct effect calculated, and those two together are our total negative effect. So the challenging here is really how we can estimate those two high dimensional parameters. So we want to propose some novel prior, but some prior that is proper and nice. So we recall what properties those coefficients are expected to have. One is they're expected to be sparse, as we said, and spatial dependency because they are corresponding to images. And another additional thing we want to address is really the concurrence between these two. Because only if both these two are not zero, we will have our mediation effect. One of them being zero, they will not enable this pathway. So we are particularly interested in the case where both alpha and zero are non-zero here. So we propose a Bayesian hierarchical prior. The idea is actually simple. It's on the first level, we want to have some variable selection indicator to select which pixels are predictive non-zero. When we're selecting non-zero pixels, what we want is we want it to select a region instead of some single pixels. That's why we will use the Ising distribution, which will promoting we selecting a continuous region instead of just one single pixel. And so in this step, in this first level, we select the non-zero pixels. On the second step, we know in this area, those coefficients are non-zero. We will estimate their value, their effect size. And we also want to add some additional smoothing to it because we don't want the effect of our non-zero coefficients to be quite noisy, which may looks like they're multicollinearity or so. The first level, we have kind of spatial variable selection, and then on the second level, we have some smoothness on the non-zero coefficients. So for doing so, on the first level, we propose, yeah, as we said, the Ising prior. So the Ising prior has some hyperparameters. One hyperparameter is for control how sparse it is, and another hyperparameter is control how strong. Like suppose this pixel is being picked, we want to pick its neighbor as well, so that there will be a region, right? That how strong we want to encourage those type of continuous selection. So those B parameters are for this. And particularly this capital B alpha beta, it's for encouraging the concurrence of our alpha and beta, which will enable our pathway. And then on conditional on our variable selection indicator equals to 1, which means we have non-zero things, we further propose our Gaussian Markov-Randall field prior, which is for adding smoothness. I think this step is kind of more natural. So we will set the mean at the averaging of the particular voxel and its neighborhood, and we will assign it a small variance, where the variance will get even smaller if there are more neighborhood, because we feel there will be more information if there's more neighborhood that we can look at together. So this is the idea of this kind of new prior. So this page is for talking about the causal effects. So because we are talking about treatment, association will not make a difference, because if we detect those regions, but the relationship is only association, not causal, it means if we protect this region, we will not guarantee to have better outcomes. This difference of causal and association, right? So we attempt to have some causal results, which is very favorable. We tried our best. In short, what we do is here we adjust for the confounders as reasonable as we can to fulfill those causal assumptions. So the idea is like this, but I'll skip the details because I feel there's a lot of debate in the causal conclusions. So I'll just skip this page. But the idea is we try to address the confounders and we care about the causal effects. So we did some simulation in the two-dimensional space. So here we show the two parameters are beta, alpha, and alpha beta multiplied together in 2D space. The simulation is quite nice. Here's our estimates, and here's the estimates from our competing methods. And also from the RMSE, our method looks quite nice, especially for the complicated true value shapes like butterfly. For the squared, the low rank method still works well because they're in nature low rank shapes. We also did some 3D simulation because our real data is in 3D, and it also works quite well. And here's our real data. This is our trial SGMB03. We deleted all the missing data so that we ended up with only 61 sample. Hope we can improve this, but for this current project, we deleted all the missing data. This is a small sample size given our model, right? But this is still a nice sample size if we're thinking this is a pediatric cancer trial. Usually their sample size is small. So our treatment is the risk groups, 1 coded for high and 0 for average. Our mediator is our 3D brain image, which is a kind of a cubic, but it only has those valid number of voxels. I shouldn't say only because it's quite large. Our outcome is the working memory standard score change from the first year to the end of the third year. So see the development during the third year period of the kid. The confounder we adjust are those. So these are the confounders that our collaborators and feel important also from the early generation of those trials, kind of using the experience identify those confounders, and we applied our model to this real data. And here's the estimated alpha parameter. most of them are negative, colored in red. It means the radiation therapy damaged the brain in those regions. And those are the beta parameter, which means better brain condition in those regions will give us better working memory development. And here's the brain region to the most of our interest, because these are the region that we have both alpha and beta being non-zero. Those are regions that enable our negative pathway. And you can see the effects are red, and most of them are negative. So our hypothesis is if we protect those regions during the radiation therapy, in theory, we will benefit our children with better working memory development in 3 years. And we can see slightly over 50% of the negative effect from treatment to the working memory is explained by our brain mediator, but there are another 50% that remain unexplained, which means we need to study more on this case. And our collaborator was able to identify some particular brain regions that they found interesting and interpretable. So that's about this project. It's a more statistical project, and I really liked it. So that's kind of the two major projects I hope to share.